start the session now. Okay, today's topic is an interesting topic, that is a webinar on novel therapeutic approaches for antimicrobial resistance. So now the introduction. First we are going to see the introduction. In the introduction, the antimicrobial resistance. So what is antimicrobial resistance? Antimicrobial resistance occurs when microorganisms, that is such as bacteria, viruses, fungi and parasites and protozoans, these may change in ways that render the medications used to cure the infections they cause ineffective. So this is known as antimicrobial resistance. The antimicrobial resistance is any microorganisms enter in our body and it will our body produce antibodies against that. So in that way, this antimicrobial resistance, the microorganisms change in ways that render the medications used to cure the infections they cause ineffective. When the microorganism becomes resistant to most antimicrobials, they are often referred to as superbugs. So most of the antimicrobials were resistant to the microorganisms such as fungi, bacteria, parasites, and they are all superbugs. And also in particularly Pseudomonas bifida bacteria is also called as a superbug and because it has the capability to degrade the oil. So that particular bacteria also referred as superbug. So like that, here the microorganism becomes resistant to most antimicrobials. They are often called as superbugs. And this is a major concern because a resistant infection may kill and also can spread to others and poses huge cost to individuals and society. Antimicrobial resistance is a broader term. So it is one of the broader terms for resistance in different types of microorganisms. So this antimicrobial resistance is resist for different types, not only particular for bacteria or fungi or virus. It will resistant in different types of microorganisms and encompass that antibacterial, antiviral, antiparasitic and antifungal drugs. These all the drugs are resistant to this. These all are called the antimicrobial drugs. It encompasses the resistance of the microorganisms. And this antimicrobial resistance occurs naturally, not it will occur artificially. It will occur naturally, but it is facilitated by the inappropriate use of medicines. So it will use by inappropriate condition of the medicine. By using the medicines or inappropriate use of the medicine, it will occur naturally. This antimicrobial resistance will occur naturally in our body. And this it is today one of the most important public health problem, is it? So nowadays also common that disease is the worldwide is the world is what they can say, the world is full of with COVID-19. You all know, is it? So for that also still now they are not discovered any confirmation of that vaccine. So that or the antiviral vaccine, they will discover or develop the antiviral vaccine against that COVID-19. But this antimicrobial resistance is today one of the most important for the public health problem and is destined to become one of the main health challenges of the coming decades. And the increasing incidence of these infections with multi-resistant microorganisms. So what is multi-resistant microorganisms? The microorganisms will be resistant for multi-drugs. So more than 2 drugs, the microorganism will resist. That is multi-drug resistant organism. That it dramatically reduces the therapeutic equipment that is available in the doctors, and it leads to an increase in the complexity of treatments with an increasing, that is morbidity, morality and cost associated with the health care and generates social alarm that is difficult to manage in that situation. So this is about the antimicrobial. So it is one of the most powerful tool, that is it is one of the most important The next one is this antimicrobial multi-drug resistant organism. For example, a doctor prescribes an antibiotics for the treatment of any bacterial infection means it will not affect any against viruses. So it will only affect against that particular bacteria, not against the viruses. So note whether an infection is bacterial or either viral helps to effectively treat it. If people overuse that antibiotics or use them incorrectly, the bacteria might become resistant. So this means that the antibiotic becomes less effective against that type of bacteria as the bacterium has been able to improve its defenses. So the doctor can prescribe antibiotic to treat a wide range of infections. A narrow spectrum antibiotic is only effective against a few types of bacteria. So some antibiotics attack the aerobic bacteria, not while others against the anaerobic. So it only particularly attack the aerobic, not the anaerobic bacteria. This aerobic bacteria means they need oxygen, and the anaerobic bacteria they do not need any oxygen content. Then these antibiotics are powerful medicine that fight certain infections and can save lives when used properly. They either stop bacteria from reproducing or destroy them. So these antibiotics, now we are going to see about the antibiotics. So resistance is entirely different from the antibiotics. Now we are going to see the antibiotics. These antibiotics are powerful medicines that fight certain infections and can save lives when used properly. They either stop bacteria from reproducing or destroy them. So, for example, if we are affected by any infection, suddenly we will go to with the doctor, and we will get an antibiotic. The doctor will prescribe us some antibiotics, and we are taking the antibiotics. But if that antibiotics only can stop the bacteria from reproducing or for their multiplication. Once our body get that antigen, that is bacteria, it will suddenly replicate, or it will take multiplication will take place. Multiplication of bacteria will take place in our body, and it will either stop bacteria from reproducing or destroy them. That is antibiotic. Before bacteria can multiply, can cause some symptoms. So first, if in our body the bacteria enters, the bacteria suddenly enters our body, and it will occupy in some of the region or organs of in our body, and it will take multiply, and after that multiplication only it causes the symptoms, and after the symptom only our body will produce our immune system will produce the antibodies, and it will typically kill them. So in our immune system, that white blood cells attack harmful bacteria, and even if the symptoms do occur, the immune system can usually hope and fight off the infection. So this is an antibiotic. Sometimes, however, the number of harmful bacteria is excessive, and the immune system cannot fight them all. So, number of bacteria, not a single bacteria, a number of bacteria enter in our body, and our immune system can't fight them. Why? Because there is a more number of bacteria inside in our body. So it can't fight against. So we need antibiotics. So these antibiotics are useful in this scenario. So for that option only, we are using the antibiotics. And the first antibiotic was penicillin. We all know, I think you all know the first antibiotic was penicillin, and this penicillin was obtained from a fungi called Penicillium notatum, and it was first discovered by Alexander Fleming in 1942, and he got the Nobel Prize in 1945. So, and this penicillin-based, some other penicillin-based antibiotics are available, such as ampicillin, amoxicillin, and penicillin G are still available to treat a variety of infections and have been around for a long time. So these are the common things about the antibiotics. So mainly we have to know whether the antibiotics and the antimicrobial resistance is entirely different. Then next one, how do antibiotics taking the antibiotics and how it will works is it. So there are different types of antibiotics are there which work in one of two ways. First one is the a bactericidal antibiotic and second one is the bacteriostatic. So this bactericidal antibiotic such as this penicillin, if it kills the bacteria, so if any bacteria present in our body and also we are taking the bactericidal antibiotic, it kills that bacteria, and these drugs usually interfere with either the formation of the bacterial cell. So that antibiotic immediately kill the bacterial cell wall and it destroy the cell content of the bacteria. So the bacteria is harmless to our body. The next one is the bacteriostatic. This bacteriostatic, which stops bacteria from multiplying. So this bacteriostatic antibody is it is used to stop the multiplication of the bacteria. So not to kill or anything. So it will only arrest the multiplication. These are the two different types of antibiotics which work in one of the two ways. And next one is the viruses. Viruses cause most upper respiratory tract infections, that is such as common cold and flu, and these are the viral common diseases. And here the antibiotics do not work against these viruses. Only this antibodies helps to destroy the bacteria, not for the viruses. Okay, and next one is here this antibiotics are next slide. Now these are the main difference between the antibiotic resistance and the antibiotics. Now we are going to see about the antibiotic resistance types. So in this antibiotic resistance, there are two types of there. That is natural upward. Okay, so there are two types: natural antibiotic resistance and the upward are there. In this natural, here the lack of metabolites are the process of targets regular, and this upward there are two types, that is genetic method and biochemical mechanisms. In the genetic methods, there are two methods are there: chromosomal methods and extra chromosomal. So the chromosomal methods will occur through the mutation. The sudden change occurs in the gene. It will take in the chromosomal method, and the extra chromosomal method here the plasmid. So plasmid actor it is an extra chromosomal. So in here some genetic changes will occur. That is the genetic method. And in that occur another one, that is biochemical mechanisms. In the biochemical mechanism, here some methods are there, that is by producing enzymes and preventing drug accumulation, monitoring target site, and use alternative paths. So these are the biochemical mechanisms for the antibiotic resistance. Now the natural. This natural antibiotic resistance is otherwise known as intrinsic resistance. It is also known as intrinsic resistance, and it is the innate ability of the bacterial species to resist the activity of a particular antimicrobial agent through inherent that structural or functional characteristics, or allowing tolerance to a particular drug or the antimicrobial class. This natural resistance, that intrinsic mechanisms or this resistance are the innate characteristics of the microorganism and are transmitted to the protein vertically during the cell division. For example, anaerobes are intrinsically resistant to the aminoglycosides. So this aminoglycoside is an antibiotic, and it is intrinsically resistant to the anaerobe bacteria. The next one is the upward resistance. This upward resistance, this mechanism of resistance are caused by changes in the usual genetic changes, that is makeup of microorganisms and the results of altered cellular physiology and the structure. So it will, some alteration may take place in the cellular cell and also the structure. These bacteria develop resistance due to the mutation following exposure to the antimicrobial agents or receive resistant properties passively from the resistant organisms. For example, we can say that the methicillin resistant to the methicillin antibiotics resistant to Staphylococcus aureus bacteria. These are the natural and upward antibiotic resistance. Then the next one difference between antibiotic and the antimicrobial resistance. So in this, here we are going to see about the differences. The antibiotic resistance occurs when bacteria change in response to the use of antibiotics used to treat bacterial infection, that is such as urinary tract infection, pneumonia, or bloodstream infections, making them ineffective. So this antibiotic resistance is only for the antibiotic is used for only the bacterial infections, but this antibiotic resistance occurs when bacteria change in response to the body. And the antimicrobial resistance is a broad term encompassing resistance to the drugs that treat infections caused by other microbes as well as such as parasites, that is parasites which cause the disease like malaria or helminths, viruses like some HIV, fungi, candida. So these all are the diseases caused by the microbes, and it is encompassing the resistance to drug resistance for the to drugs. And advancement of the effective means of the control and rapid detection is essentially for the treatment and prevent of multiple bacterial infections, not only the single bacterial infections, and prevent of multiple bacterial infections resistant to the antibiotics. Many control systems including combination therapy, phages, and systems based on the antibiotic resistance mechanism, that is a flex pump and beta lactamase inhibitors. This will study elaborate and have been designed to improve the antibacterial activity against multiple antibiotic resistant pathogens. And another one difference is that different approaches have been developed for rapid, selective, and sensitive detection for multiple antibiotic resistant pathogens, including nucleic acid probe, chemical methods, physical methods, and molecular based techniques. So these are the common difference between the antibiotics and the antimicrobial resistance. And also the novel therapeutic approaches such as a survey of different alternatives in development to conventional antibiotics using antibodies for the treatment of Staphylococcus aureus biofilm. So this Staphylococcus aureus biofilm is the antibody is conventional antibody. We are using that conventional antibody and developing immunocommensurate therapies. And also the bacteriophages. The bacteriophages naturally target a specific bacteria, and the antimicrobial resistance has required interest in using them therapeutically, including through that is bacteriophages to act as a biologic source using temporary phages. So here antimicrobial resistance can also occur in viruses to antivirus, and this issue ends with an infographic on the current direct acting antivirus usage. The antivirus used to treat the hepatitis C virus and the development of antiviral resistance. So it will develop the antiviral resistance. The bacteriophage acts as the antiviral resistance. And next, the antimicrobial resistance can also occur in viruses to antiviral resistance. And next, we are going to see the new methods that have the potential to deliver the novel antibiotics. So here, the bacteria produce antibiotics that kill or inhibit the replication of the competition. So in the new methods, the bacteria will produce the antibiotics, and it will kill or inhibit the other organisms for the replication of the competition. So here today the marketed antibiotics such as streptomycin have been derived from bacteria that grow on artificial solid or liquid media. However, most species of bacteria will not grow on artificial media. Only it will grow on the in the natural media. So here the streptomycin has been derived from the bacteria. It grow only in the artificial media. The next one. Here the marketed antibiotics have been isolated from the non-culturable bacteria, since growth on solid media has been as an essential step to the development antibiotics. So it is possible to clone large fragments of non-culturable bacterial genome, and it will fragments the non-preserved bacteria, fragments the bacterial genome and to express them using recombinant DNA or DNA technology. It will using to recombinant DNA technology. Then next we are going to see about the mechanism of antimicrobial resistance. So here the bacteria have evolved sophisticated mechanisms of drug resistance to avoid the killing by antimicrobial molecules, a process that has likely occurred over millions of years of evolution. So here the most relevant mechanism here, for example, fluoropurin resistance can occur due to three different biochemical rules, all of which may exist in the same bacteria to prevent it, and it will produce an additive effect and often increasing the level of the resistance. It will increasing the level of the resistance. Here some chemical alterations also may take place. Chemical alterations of the antibiotics also may take place in the mechanism. The production of enzymes. So the production of enzymes is capable of introducing some chemical changes to the antimicrobial molecule. It's a well-known mechanism of upward antibiotic resistance in both gram-negative and gram-positive bacteria. So it is the upward antibiotic resistance for the both positive and the gram-negative bacteria. And interestingly, most of the antibiotics affected by these enzymatic modifications exert their mechanisms of action by inhibiting protein synthesis at the ribosome level. So many types of modifying enzymes have been described, and the most frequent biochemical reaction they catalyze include, that is acetylation. The acetylation includes aminoglycosides, chloroamphenicol, streptogramins. So these are the acetylation catalysts, enzymatic catalysts. And phosphorylation. This phosphorylation includes the aminoglycosides and chloroamphenicol antibodies. And adenylation includes adenylation, aminoglycosides and glucosamides. These are the steps in the adenylation process, that enzymatic reaction. And regardless of the biochemical reaction, the resulting effect is often related to steric hydrogenase that decreases the ability of the drug for its target. So which in turn is reflected in higher bacterial and the higher bacterial minimum inhibitory concentration. So the minimum inhibitory concentration is a higher. And it is the most relevant mechanisms giving examples that have relevant clinical impact are here. The first step is the modification of the antibiotic molecule, decreased antibiotic penetration and efflux, changes and or bypass of target site, resistance due to global cell adaptive process. These mechanisms, these most relevant mechanisms will take place. And first here, that is limiting uptake of a drug. Sorry. Limiting uptake of a drug. So there is a natural defense. Here the no artificial defense. Here the natural defense is the ability of bacteria to limit the uptake of antimicrobial agents. The structure and so we can see the gram-negative bacteria. The structure and function of the gram is so the lipopolysaccharide layer is provides a barrier to certain types of molecules. And this bacteria modify their cell membrane. So this gram-negative bacteria modify their cell membrane, the foreign channels occur, and thereby preventing the antimicrobials from entering into the cell. So there are two main ways in which foreign changes can be. That the drug uptake increasing the number of porins present and mutations that changes the selectivity of the foreign channel. These are the two changes linked to the drug uptake. And this strategy can has been observing many gram-negative bacteria, not in gram-positive. That is such as Pseudomonas, Enterobacter, Klebsiella species against drugs such as that is imipenem, the aminoglycosides and the quinoline. These drugs have been observed in many gram-negative bacteria. That is not resistant to the gram-negative bacteria. And the gram-positive bacteria, that is Staphylococcus aureus, recent similarly has developed a resistance to the vancomycin. So this vancomycin is resistant to Staphylococcus aureus, that is gram-positive bacteria. This Staphylococcus aureus produces thickened cell wall. So also we know when we are doing the gram staining, there gram positive and gram bacteria, we can identify the gram positive and gram negative bacteria, and that gram positive bacteria containing peptidoglycan layer and the gram negative bacteria containing lipopolysaccharide. So due to that changes here, some resistance also may be occurred. This Staphylococcus aureus is a thickened cell wall. So it contains what? Peptidoglycan layer, and it makes difficult for the drug to enter the cell and provide an intermediate. So it is in the resistance to the vancomycin antibody. The next one is the modification of a drug target. There are multiple components in the bacterial cell that may be targets of antimicrobial agents, and there are just as many targets that may be modified by the bacteria to enable resistance to those drugs. One mechanism of resistance to the beta-lactam drug is almost exclusively by gram-positive bacteria. So here the resistance to the beta-lactam drugs, it is exclusively resistant to the gram-positive bacteria through the alteration in the structure. So the structure is, there are some changes may occur in the structure of the gram-positive bacteria or the number of the PBS. And with this PBR transcriptase and involved in constructions of peptidoglycan in the cell wall. So the changes in the structure may decrease the ability of the drug to bind or totally inhibit the drug binding. It is the modification of drug target. And the next one is the inactivation of the drug. So the inactivation of the drug, there are two main ways in which bacteria inactivate the drugs. First one is the actual degradation of the drug, and the second one is the transfer of a chemical group to the drug. So in the actual degradation of the drug, here the beta-lactamases, so that is the beta-lactamases are very large group of drug hydrolysing enzyme. So this beta-lactamases are a hydrolysing enzyme, drug hydrolysing enzyme, and it is a very large group. Another drug can be inactivated by hydrolysation is tetracycline through the TEX gene. Here some alteration of genes is taken, that is mutation. And the next one is the transfer of chemical group to the drug. Here during the inactivation by transfer of a chemical group to the drug, most commonly uses that transfer of acetyl. So in this, transfer the acetyl group in this drug, it will transfer the acetyl group, and there are large number of transferases that have been identified, and here the acetylation is the most diverse. So there are more number of transferases have been identified. Among that, the acetylation is the most diversely used mechanism and is known to be used against the aminoglycosides. So and it is most used to the aminoglycosides. So these are the main inactivations of drug, two types. And next one is the active effects of the drug. Here the active effects of a drug means the bacteria possess chromosomal encoded genes. So here it is, this bacteria possesses an encoded gene, that is chromosomally encoded gene for efflux pumps are here. So here in that pump, in that pumps, efflux pumps, some are expressed constitutively and others are induced or it is overexpressed. It may be induced or overexpressed under certain environmental stimuli, or when a suitable substrate is present. When a suitable substrate is present. The effluent pump functions primarily to rid the bacterial cells of the toxic substance. So here the effluent pump, it will get rid of the bacterial cells of toxic substances, and many of these pumps will transport a large variety of pumps. So it will transport the large, not only small amount, it will transport a huge amount of compounds through that pumps. And this figure represents that efflux pumps in gram positive and gram negative bacteria. So here some that ABC superfamily is there, and that ABC superfamily, the new first is that numerous drugs, and then that MFS family, there is acrylamine, benzethylene, citramide, chlorhexidine, pentamidine. So these are the things are there. And in MABE family, so here more families are there in the gram positive. First one is this is the super family. MFS, MABE family is the mark. So here it will take place in the cytoplasm. And this MABE family, the aminoglycosides and chlorophenols carrying drugs are there. And in the SMR family, acrylamine, benzodiazepine and citramide. And these all the things are present in the gram positive, and in the gram negative, here also the three terms are there. That is OR and D family, ABC superfamily, and MFS family. So here the numerous drugs are there coded there. That is coded. And in the ABC superfamily, that macrolides and MFS novoboracil. These are, and also nalidixic acid. And these are occurs in the cytoplasmic region of the gram positive and gram negative bacteria. And here five major families of the eclipse comes including the major facility superfamily, that is a major MFS means major facility superfamily, and SMR the small multi-drug resistant family, the resistant no-deletion cell division family, that is RND, and the ATP binding cassette family, that is ABC, and the multi-drug and toxic compound 60, that is MATE. MAT is the multi-drug and toxic compound exclusion family. So these families differ in terms of structural conformation, energy source, range of substrate they are able to exclude, and in the type of bacterial organisms. And this active effects of drug, here the most of the bacteria possess many different types of effluent pumps. There are five main families that we have discussed, that is ATP binding cassette, multi-drug tox compound, small, that